Hi there, and welcome to my talk on insulin-stimulated glucose transport in health and insulin resistance in adipose tissue and muscle. Who am I? My name's Daniel Fazakerley. I'm a new PI at the Institute of Metabolic Sciences in Cambridge, and I'll just give you a brief background to how I got to here. So I did my PhD at the University of Bath in the UK with Professor Jeff Holman, and I studied GLUT4, glucose transporter trafficking, specifically GLUT4 trafficking in muscle cells, and that was 4 years from 2006 to 2010. After which I moved to Sydney in Australia to work with Professor David James on a Henry Wellcome postdoctoral fellowship. And while I was in Australia, I was first at the Garvan Institute and then at the University of Sydney. And as you can see from the timeline there, that was a fairly extended postdoctoral stint. But in 2009, I got the opportunity to return to the UK with support from the MRC as a principal investigator to set up my own group at the Institute of Metabolic Science. And basically throughout my career, I've been interested in how insulin causes glucose transport to increase in adipose tissue and muscle cells. And so that's what my lab works on now. We study the cell biology of insulin-stimulated glucose transport and how this goes wrong in disease. Okay, so just a brief overview of my talk. I'll give you a bit of a history and the underlying principles of the cell biology of how insulin stimulates glucose transport. Because of the topic of the course we're on, I'll focus a little bit on muscle and how this works differently in muscle to adipose tissue. Give a brief overview of what's known about the importance of GLUT4 in whole body metabolism, and then talk a little bit about our work, more recent work, on what causes insulin resistance in adipose tissue and muscle. Okay, to start with the kind of overview of the cell biology. This is a figure from a review that I wrote with Jacqueline Stockley and David James in 2011. And basically what we did is we put together a timeline of the key events in the history of studying GLUT4, insulin-stimulated glucose transport. And as you can see, back in the 1930s, it was observed that insulin-stimulated glucose transport into muscle. And these early experiments really showed that it was the transport of the glucose into the muscle that was a rate-limiting step for glucose usage by muscle in response to insulin. But it wasn't clear how this occurred. Was this because there wasn't a process that was activated by insulin? For example, there was a transport of glucose that was targeted by insulin signaling, and this would increase its activity? Or was there a change in the composition of the plasma membrane, for example, so that a glucose transporter wasn't there and in response to insulin was there? And it turns out that it took into the 1980s, the translocation hypothesis was confirmed by experiments by Sam Cushman's group, where they showed that basically what happens in response to insulin is if insulin's absent, the insulin-responsive glucose transporter is actually sequestered from the plasma membrane. And in response to insulin, GLUT4 increases at the plasma membrane, thereby facilitating glucose transport. So in the late 80s, it was found that this insulin-responsive glucose transporter, whatever it was enriched in muscle and fat tissues. And then by the end of the 1980s, it was actually cloned. We found the gene responsible for this, and it was named GLUT4, so glucose transporter 4. And the cloning of this gene, so we had the sequence, would allow people to now generate reporter constructs that were tagged. And this really gave birth to a large increase in studies on the cell biology of GLUT4, looking at its trafficking and the regulation of its trafficking. So for example, this is GLUT4 here. It's a 12-pass transmembrane protein, and here it's depicted inside a vesicle. And you can see that we're able to make fusion constructs that have C-terminal tags, so these tags are in the cytosolic portion of the cell. And we can also even tag it in this luminal portion here, so either inside the vesicle or when the protein's in the plasma membrane, this will be accessible to the media, so outside of the cell. And we can use reporter constructs like this to do a wide range of cell biology experiments. But in particular, we can use it to assess PM contents, the amount of GLUT4 in the plasma membrane, really quickly and readily. So here I'm showing you an experiment that we did actually pretty recently. This is done in 3T301 cultured adipocytes. And on the left here, you can see the total amount of the glucose transporter expressed in the cells under unstimulated and stimulated conditions. And this doesn't really change depending on whether insulin's there or not. This is only an acute treatment, so 20 minutes with insulin. But in contrast, what you can see, if we now stain those cells with an anti-HA antibody, And what we've done is made an epitope, put the HA epitope in this part of the protein here. If we don't permeabilize the cells, this gives us a really nice way of assessing the amount of GLUT4 that's actually within the plasma membrane and accessible to glucose for glucose transport. Without insulin, GLUT4 is really effectively excluded from the plasma membrane. But with insulin, you can see this beautiful recruitment of GLUT4, and you get these really nice images that you can quantify and get a quantitative readout for the amount of GLUT4 translocated to the plasma membrane. And there's been a whole raft of other constructs made that have different features that allow us to study more specific processes of GLUT4 trafficking, and I'll show a video of this a little bit later. So following this, the cloning of GLUT4 also allowed GLUT4 knockout mouse models to be made, and I'll talk a little bit about how these have informed the field later on. But more importantly, for this part of the talk, this really marshaled in a series of studies that are really underpin our understanding of how insulin stimulates GLUT4 translocation in adipocytes and muscle cells, including the trafficking processes and some of the signaling elements as well. So I'm just going to go over the current models for how GLUT4 traffics within the cell, and then go on to how we think the signaling pathways intersect with this trafficking process. So GLUT4 is an integral transmembrane protein, so it's co-translationally inserted into the ER membrane, where it's trafficked through the secretory pathway. Now, if this was a constitutively secreted protein that's delivered to the plasma membrane, it would leave the trans-Golgi network and to be delivered straight to the plasma membrane. But GLUT4, as I've shown before, is actually sequestered within the cell in the absence of insulin. So this is an example where insulin is absent, and it's actually trafficked into this specialized compartment here, which I've termed GSV, which stands for GLUT4 storage vesicle, or IRV, which is insulin responsive vesicle. And in the field, these are actually used relatively interchangeably, or can lead to some confusion. But what these basically are is a compartment of small vesicles, so only 50 to 100 nanometers. They're where GLUT4 is mainly sequestered in the cell in the absence of insulin. They're highly insulin responsive. So these are the part of this population of GLUT4 are mobilized in response to insulin. So if we now add an acute insulin dose, what happens to these vesicles is they're trafficked towards the plasma membrane, and insulin acts to basically release them from this storage, allow these vesicles to traffic to the plasma membrane, which is likely aided through elements of the cytoskeleton. These vesicles are then insulin also promotes their interaction with the plasma membrane, likely promotes their fusion with the plasma membrane, and there we have increased amount of GLUT4 at the plasma membrane here. We can actually visualize this using a GLUT4 reporter. So this GLUT4 reporter is slightly different from the one I talked about earlier, where we had an HA epitope in that luminal portion. This one has a pH-sensitive GFP variant, and this is an adipocyte expressing these. And what you're going to see is that when GLUT4 is delivered to the plasma membrane in these small vesicles, the pH change allows you to see a flash of an increase in fluorescence. Okay, so I just start this video. We're going to zoom in over the cell, and you can see these flashes of green, and that's the delivery of GLUT4 in small vesicles to the plasma membrane. So we can actually visualize this process, the delivery of GLUT4 to the plasma membrane, using specialized constructs. And I guess I can make the point here that these different constructs allow us to really monitor different aspects of the GLUT4 trafficking process. So we can actually monitor these fusion processes, zoom in on them, look at the time they take, etc., like that, and get real detailed information on specific parts of the GLUT4 trafficking itinerary. Okay, but of course, the delivery of GLUT4 to the plasma membrane is not a one-way process. It's also internalized into the endosomal system. But insulin may also act here, and there are some studies suggesting that insulin slows the internalization of GLUT4. Another way it will promote GLUT4 accumulation of the plasma membrane. And also, as cells are continuously incubated in insulin and GLUT4 is internalized, insulin is also known to promote recycling from endosomes to the plasma membrane. So the point here really is that insulin targets multiple trafficking processes in the itinerary of GLUT4 to promote accumulation at the plasma membrane and facilitate glucose transport. Now, obviously, I've just presented quite a lot of years of work in a very simple model, and I've just listed some of the labs, there are probably others as well, that have contributed to our understanding of how this process works. Okay, so that was how GLUT4 is trafficked, but what are the signaling intermediates involved in this? So insulin signals via the PI3 kinase AKT pathway to lead to the activation of AKT. Now, activation of AKT is required for insulin-stimulated GLUT4 translocation. And the main downstream substrate of AKT is this protein here called TBC1D4 or AS160. This protein is a Rab gap protein, so it inactivates Rab proteins. And so that any Rab protein residing within the GSV compartment or on the GSVs, which would normally promote trafficking to the plasma membrane, is retained in an inactive state. Phosphorylation of this protein by AKT inactivates it. So this now allows the Rab proteins on the GSVs to become GTP loaded and active and promote GLUT4 trafficking to the plasma membrane to promote glucose transport. Now, these are key signaling events in this process. If you inhibit AKT, insulin can no longer stimulate GLUT4 translocation. And if you express a dominant negative TBC1D4 that can't be phosphorylated, this completely blocks insulin-stimulated GLUT4 translocation. But there are other sites, as I indicated in earlier slides, where insulin intercedes in GLUT4 trafficking. For example, by activating RAC, PAC, and RAL-A proteins, insulin signals the cytoskeleton to promote GSV trafficking to the plasma membrane. And also, we know from a number of studies that there's likely to be key insulin signaling events occurring at the plasma membrane to control the interaction of the GSV with the plasma membrane and its fusion. And for example, there is indication that insulin may result in or change the phosphorylation status of key members of the snare complex, which is a key complex in mediating the interaction between the vesicle arriving at the plasma membrane and the plasma membrane. But there are likely other steps here that we don't yet know and key signaling intermediates that we haven't identified. Okay, so I'd like to focus a bit more on muscle specifically now, given the title of our seminar course. Now, the majority of the studies that have focused on GLUT4 trafficking that I've just described to you have been performed in adipocytes. And the simple reason for this is a much more experimentally tractable model for many reasons, including the fact that we have very good cell culture models that allow really nice cell biology trafficking experiments to be done. In contrast, in muscle, we don't have such good cell models. And although we and others have used the L6 myotube model shown here as a model as a muscle cell model for studying GLUT4 trafficking, this is somewhat limited because they don't respond to insulin quite so well. They actually have a limited endogenous GLUT4 expression. So this anti-GLUT4 I'm showing you here, this is a cell overexpressing GLUT4. So it's not clear whether they actually have the right compartments that would be present in a muscle tissue to store GLUT4. Muscle tissue and muscle cells are more difficult to fractionate than adipocytes because they have a lot of contractile apparatus, which can make that more challenging. And they also lack key structural elements of real muscle, such as T-tubules. And this might be very important because whereas the cell surface in adipocytes is purely the plasma membrane, so the trafficking of GLUT4 is from within the cell to the plasma membrane, the limiting surface. In muscle, this is different. You have the plasma membrane or sarcolemmal membrane, but you also have T-tubules. I'm showing you an image here of another GLUT4 reporter. This time it's got a GFP tag in the cytosolic part of the protein. And I just want to zoom in on these images here where we have basal or insulin stimulated. And these are cardiac myocytes, actually. They're not striated muscle, but they're cardiac myocytes. What you can see here is that under basal conditions, the GLUT4 within the cell is kind of more randomly distributed. When you add insulin, it kind of lines up along these lines here. And this is actually accessible to antibody, as you can see here from the red, which is an anti-HA Fab fragment. And you can see when we quantify this, these line up almost perfectly, suggesting that this GLUT4 is accessible to the external medium and can mediate glucose transport. And what we think this is GLUT4 within the transverse tubular network membrane. So in muscle, GLUT4 trafficking occurs to both the plasma membrane, but also the T-tubular membrane as well. In addition to this difference, there are other unique regulators of GLUT4 trafficking that are specific to muscle and fat. Now, many of the early studies that I've kind of brushed over here to generate this model, the assumption was made that much of the machinery would be the same between adipose tissue or adipocytes and muscle cells. And I think this largely holds, but then there are some specifics. So for example, the RABs that are localized to GSVs appear to be different between adipocytes and muscle cells. And in addition, there are specific proteins that are localized to the GSV compartment, namely TRAG1, which is a adipose-specific protein that we're particularly interested in at the moment, or this other RAB gap, TBC1D1, that's specifically localized to GSVs in muscle. So clearly there's a need to try and understand muscle, although many of the features of insulin-stimulated glucose transport are likely similar between adipose tissue or adipocytes and muscle cells, there are clearly some key differences. And one of the major differences is that in muscle cells, additional stimuli can also promote GLUT4 translocation to the plasma membrane. So in addition to insulin, contraction and exercise stimuli also cause GLUT4 translocation to the plasma membrane. Now, the signaling intermediates behind this are not yet fully resolved, but it's thought that, again, RAC and PAC may play a key role in this, activation of these proteins and mediation of the cytoskeletal elements. And also the energy sensor AMPK is reported to promote GLUT4 translocation, likely through targeting and phosphorylating the RAB gaps, TBC1D4, or more prominently TBC1D1, which again, as you recall, is a protein that's more unique to muscle. There's a couple of other interesting features here. So for example, if you stimulate muscle with both insulin and contraction or AMPK activation, you get an additive response. So you get more GLUT4 than with either stimulus alone, which is mechanistically quite interesting because it suggests that maybe these target different pools of GLUT4, or they invoke different mechanisms for promoting GLUT4 accumulation at the membrane, and thereby resulting in a larger increase in GLUT4 translocation. The other really interesting thing, more therapeutically, is that contraction or AMPK-stimulated GLUT4 translocation remains intact, even in insulin-resistant muscles. So in these muscles, insulin won't increase GLUT4 translocation to the same extent as in insulin-sensitive muscles, but these pathways, contraction, AMPK-stimulated GLUT4 translocation, remain intact. So understanding the basis of these pathways may be therapeutically useful. So given these differences between adipose tissue and muscle, I think it's really important that we develop ways to study GLUT4 trafficking in muscle in more detail, whether this be through new ways of studying trafficking in detail in vivo, or through the development of new muscle cell lines that we think more accurately recapitulate what will be going on in vivo. Okay, so moving on away from the cell biology and more towards a whole body physiology, what role does GLUT4 play in whole body metabolism? So this is a fairly generic standard slide on what happens when your blood glucose rises. So in the fed state, the blood glucose increases, the pancreas detects this and secretes insulin, and then the insulin circulates to its target tissues. In the liver, it suppresses glucose output, so it stops glucose being delivered into the circulation. But obviously, it needs to lower the blood glucose, so it does this by targeting the GLUT4 expressing tissues, adipose tissue, and muscle. And this is the GLUT4 part of the equation over here. And just to point out that in the postprandial state, because of the amount of muscle there is, the majority of glucose, nearly 85%, is taken up into muscle tissue. Now, the importance of GLUT4 to the overall process was really highlighted nicely by a number of knockout mouse models that were generated. These showed that if you delete GLUT4, you now result in major disturbances to whole body glucose homeostasis. So, for example, the development of insulin resistance, and in some cases, the development of diabetes. So insulin-stimulated glucose transport via GLUT4 in muscle and fat, and they've done the tissue-specific knockouts as well, are both really important for whole body glucose homeostasis and maintaining glycemia. Conversely, if you overexpress GLUT4, even quite a small amount in these specific tissues, that helps glycemic control. So these mice become more insulin sensitive, and it can prevent the onset of insulin resistance, or delay it at least. And moving on to more specifically talking about type 2 diabetes and insulin resistance, there is evidence that insulin-stimulated glucose transport into muscle is an early event in the progression to type 2 diabetes. So in terms of, the insulin resistance in muscle is an early event in people progressing to type 2 diabetes. But interestingly, there's currently no treatment for type 2 diabetics that targets restoration of glucose transport into muscle. Now, this is glucose transport. Of course, we presume this means that insulin-stimulated GLUT4 translocation is impaired in these muscles because GLUT4 trafficking is rate limiting for glucose transport. But I just note, and to my knowledge, this actually hasn't been measured in in muscle, hasn't been specifically measured in insulin-resistant humans. But I just wanted to talk a little bit more specifically about what I mean about insulin resistance. Okay, so insulin resistance is where the insulin no longer works as effectively on its target tissues. So thereby it can't sufficiently decrease glucose output from the liver, and it can't sufficiently increase glucose transport into adipose tissue and muscle. At the cellular level, which is what I'm interested in, what we mean is that insulin can't increase GLUT4 at the plasma membrane to the same level as it can in an insulin-sensitive cell. So this means that either insulin signaling is impaired, or the for some reason, the trafficking of GLUT4 to the plasma membrane is impaired, or the generation of the insulin-sensitive pool of GLUT4 is impaired. And I've put all these crosses here because really, it's not clear where insulin-stimulated GLUT4 translocation breaks down in insulin resistance. This is a major question in the field. Is it a signaling impairment? Is it a trafficking impairment? And this really isn't clear. However, the majority of studies, and the studies I'm going to show you that we contributed to, don't focus on this. And I think this is probably something that can be done now with the tools at hand. But what I'm going to talk to you a little bit now is what the upstream causes of insulin resistance in adipose tissue and muscle are. And many of the studies have focused on the idea that we know that environmental stresses are a key inducer of insulin resistance. There are genetic components as well. We know that overeating, caloric excess, and obesity are associated with insulin resistance. And the idea is that these will be translated to cellular insulin resistance via an intracellular stress pathway. And a number of these have been proposed, but the most commonly cited ones are probably lipotoxicity. So this is the accumulation of toxic lipids within cells, for example, ceramides or DAGs, or changes in mitochondrial function, namely mitochondrial dysfunction, or the increased mitochondrial oxygen production. And in some way, the accumulation of these stresses within the cell interferes with either insulin signaling or GLUT4 trafficking. Now, my work, my final work during my postdoc with David James' group was really focused on the idea of the role of mitochondrial oxidants in insulin resistance. Now, there have been a number of papers, 3 key ones of which are shown here, that have shown the association between insulin resistance and mitochondrial reactive oxygen species, or reactive oxygen species in general. More recently, it's been proposed to be more specific to the mitochondria. And that if you can target these somehow, either with a small molecule or genetically, and lower reactive oxygen species, you can improve insulin sensitivity. And we've contributed to these older papers in 2 ways. The first one was a study where we used a small molecule, a paraquat redox cycler. So this is a paraquat molecule that produces superoxide, and it's been targeted to the mitochondria by this TPP moiety. And we can give this to cells quite acutely, so half an hour to 2 hours. This goes to mitochondria and starts generating superoxide. And if we acutely induce mitochondrial loss in this way, we induce insulin resistance. We did this in both cultured cells and in tissue preparations, so adipose explants or soleus strips. And what you can see here, if you look at the open bars, is that insulin-sensitive explants respond to this extent, but in the presence of this small molecule that induces mitochondrial loss, we see an impairment of the response in both explants, in adipose explants and in soleus muscle. Now, interestingly, and I'm showing you data from the cell models here that had this same insulin resistance, this occurs without any obvious impairment in insulin signaling. So this provides us a nice model now where we can try and understand what's going on regarding the site of insulin resistance. Is this an event in insulin signaling we're not measuring, or is this more specifically a defect in GLUT4 trafficking and it's not a problem with insulin signaling? And they're studies we're going to undertake soon, I hope. And the second part of this story that I just wanted to mention was that we performed another study that associated insulin resistance and increased mitochondrial loss with loss of mitochondrial coenzyme Q. Now, coenzyme Q is an electron carrier within the electron transport chain, but also an antioxidant. And what we found was that in adipose tissue and muscle, if we isolated mitochondria, the amount of coenzyme Q within mitochondria was decreased. And this occurred after relatively acute high-fat diet feeding regimen. So this is only 5 days in adipose tissue and about 14 days in muscle. And these are approximately the times where we start to see insulin resistance in these tissues. And we performed an add-back experiment where we gave CoQ in relatively high concentrations back to the mice. And what we could do there is restore glucose disposal during a GTT into adipose tissue, which I haven't shown, and quadriceps muscle as well. So this led us to propose a kind of basic model that within mitochondria, a loss of coenzyme Q might predispose mitochondria or be an upstream driver of increased oxidants that are observed during high-fat diet feeding and other models of insulin resistance. And these increased oxidants lead to insulin resistance. But of course, the big question is, how does mitochondrial loss, so an oxidant being produced within mitochondria, lead to insulin resistance, which is a defect in a process that occurs in the cytosol? And we're hoping that we'll be able to address this by using our models of oxidant-induced insulin resistance, try and understand the effect it's having on glucose trafficking. Okay, so to summarize, insulin and exercise signaling promote glucose translocation and glucose transport in muscle via, Mitochondrial reactive oxygen species induction can acutely induce insulin resistance, and this may be promoted by lower levels of coenzyme Q in mitochondria. And one of the key questions I think we need to address is where in the insulin-stimulated glucose translocation cascade the defect lies in insulin resistance. So just to give you an idea what the active projects are in our group at the moment, this is the overview that I provided at the start of the talk. Regarding the cell biology, because this is what we're really interested in, we're trying to undertake studies to identify new regulators of glucose trafficking with a particular interest in understanding how the plasma membrane signaling events might be involved in regulating glucose translocation. We are very interested in muscle, particularly because of the knowledge that exercise or contraction-based stimuli can still induce glucose translocation in insulin-resistant muscle. And to do this, we're looking into new muscle cell lines and developing these with the idea that we can perform quite detailed cell biology experiments in a better muscle cell line. And I just alluded to in the final part of my talk is that we're really interested in using our models of insulin resistance in vitro, so in cell lines, and these oxidant-induced insulin-resistant models to try and study which aspects of this pathway are impaired in insulin resistance. Okay, so this isn't the final slide of my talk, but I just wanted to now just acknowledge my group at the IMS. We're just getting started and we're quite a small group, but thanks to Dougal and Olivia. And also the collaborators, both in my previous lab and others who have helped in studies that I published during my postdoc and I presented here today. We were asked to finish our presentation with a couple of questions for discussion. I think one that I always get asked, and I think is a really interesting question to discuss, is in insulin resistance, do you think it's a good idea to target muscle glucose uptake for treating insulin resistance? And I think when discussing this, it would be interesting to think, okay, well, it's a defect, so maybe overcoming it's a good idea. But one concept is that perhaps insulin resistance is a protective mechanism for example, if we do take the mitochondrial reactive oxygen species angle, then maybe the cell is trying to protect itself from excess mitochondrial reactive oxygen species, and promoting extra glucose transport would be detrimental to individual muscle cells. I think that's an interesting concept to think about. And if we go down that line and we think it is a good idea, how would we then try and address that? And I think one way to do that would be trying to understand how glucose trafficking is regulated by insulin and exercise signaling. One that's a bit more left field, and I haven't touched upon today at all. Now, while GLUT4 is highly enriched within muscle and adipose tissue, it is expressed in other cell types, and it actually is expressed in certain neuronal populations within the brain. And there is evidence that if you knock out GLUT4 in these neurons, you affect whole body insulin sensitivity and whole body metabolism. So I think in doing a bit of research into this, there's not a huge amount of literature on how GLUT4 trafficking is regulated in these neurons. They are insulin sensitive. Apparently leptin can also stimulate GLUT4 translocation in specific neurons. So I think it's interesting to think about how GLUT4 trafficking or insulin resistance in terms of GLUT4 trafficking in neurons may play a role in whole body glucose homeostasis. So thank you very much.